Hi everybody. Sorry I can't be there for Zoom. I would like to lead you through a little bit of a discussion of mitosis and meiosis. Now the first thing I'm going to do is I'm going to apologize for any lag that might exist during this recording, largely because I'm not working in my normal situation. But I do want to address these forms of cell division. I want to start up top and basically get an idea of how these two divisions differ from each other based on addressing the outcomes of these divisions. So mitosis, we say often, is for growth and for replacement. So if I shed some cells, like I do from my skin, I need to replace them by producing new cells that are exactly the same. Now, we can also think about the fact that, of course, if I'm going to grow from a single cell into the multicellular organism I am, I'm going to need to have that kind of growth. The result of this process is we're going to produce two cells. These two cells are identical, and they are what we refer to as diploid, or 2N. And the important thing about that is that to be diploid means that you have two copies of each chromosome. Now that'll make a little bit more sense after we address meiosis here. I'm just going to create a little bit of a dividing line between these for clarity purposes. So meiosis is all about sex. Meiosis is for sexual reproduction. It's going to produce what we call gametes. Gametes are egg and sperm. The result of meiosis is actually 4 cells. And we can see down below here that we're going to have meiosis 1 and meiosis 2. So there are 2 divisions. Well, there's twice as many divisions. We end up with twice as many cells. It's not too big of a surprise. I'm going to describe these as non-identical. Some people might describe them as being unique. But we know that anytime we're creating things through some random processes, that there is the possibility of creating identical things. It's just very unlikely given the kinds of shufflings that we're going to talk about as we go through this. And these cells, most importantly, maybe are haploid, or N. And the idea here is that we need to cut our genetic material in half. And this is for a pretty simple reason. If we think about this, N, which is an egg, and N, which is a sperm, if we add those two together, they are going to equal 2N. So it's a pretty simple kind of concept to have this kind of thing taking place. So N plus N, an egg plus a sperm, equals 2N, a diploid organism. So anyway, this is kind of what we've got going on here. I'm going to go ahead and jump into mitosis, and we can just kind of take a look and see what's going to happen in mitosis itself. Okay, let's do some mitosis. Now, it ends up that prior to mitosis, during what we refer to as interphase, we need to replicate some DNA. So I'm just going to say that first we replicate DNA in interphase. All right, and that's something we're going to have to do. We double our genetic material because in mitosis, what we want to do is to create 2 identical copies of a cell. That does mean that we need to have identical copies of the DNA as well as extra organelles that are also made during interphase. When we talk about mitosis or meiosis, those are going to be the processes by which we take this DNA and we split it up between what are going to become 2 cells. So we can start taking a look at this. Now we should be able to see here if I scroll that in mitosis, we're going to have 1, 2, 3, 4 stages we're going to talk about. Now you actually can talk about different numbers of stages, and I just want to point out we're going to have kind of the simplest view of this rather than trying to divide some of these stages into a couple of stages themselves. Now, we actually have a way to label this as well. Since we have our prophase, metaphase, anaphase, and telophase, we tend to use this acronym PMAT to remember the order that these phases occur in. Now, my question is, if I have a cell, how do I divide DNA knowing that this cell is eukaryotic, meaning that it has its DNA in a nucleus, and I need to somehow take the DNA that's inside the nucleus and put it into two separate cells? That actually kind of brings up what's going to happen in prophase. So I'm going to go ahead and write prophase off to the side, and that'll be our first stage here. And I'm going to draw a nucleus. It's a really big one. It's actually kind of the size in which a nucleus might be in certain kinds of white blood cells, like, say, lymphocytes. And I'm going to draw a really big nucleus in here, but I'm going to draw it with a dashed line, indicating that the nuclear envelope is breaking up. So that's kind of our key thing for prophase. Nuclear envelope will break up. That's going to mean that we're going to have DNA free in the cell so that it can move around. Now we need to decide what exactly we're going to have here as far as our DNA, and I'm going to draw that in a way that hopefully makes some sense. Now, in order to do this, if you haven't already got them out, you're going to need a couple of different colors. So I'm going to draw a chromosome in here. Here's a chromosome. It's a large chromosome. I'm also going to draw a small chromosome. And I've done both of those in orange. Hopefully they'll pop up in just a second. There they are. And I'm going to draw some here in green as well. So what I have is a pair of large chromosomes and a pair of small chromosomes. This is all prior to the replication. So this is really important. This is what the cell would look like, except with a nice complete nucleus during interphase, if we were able to see condensed chromosomes. Now, of course, in interphase, we actually have chromatin, this kind of uncondensed, twisted up form of the DNA, so we won't see it as individual chromosomes. Now, I want to make this look as it would if we were actually undergoing the division process. So in order to do that, I'm going to need to have these duplicates in here, and they're going to be stuck together at some centromeres. These look a little bit like K's. Sometimes we draw them kind of like X's. And this is a good point to allow us to talk about some terminology. So as we're taking a look at this, we want to say, what exactly do I see here? Well, I've got over here a large orange chromosome and a large green chromosome. These are homologous chromosomes. That is, I received, let's say, this orange one here from my father and the green one from my mother. So I have a pair of chromosomes as a diploid organism, and one from each parent. Those form homologous pairs. I have two pairs of chromosomes, the big pair and the small pair. Once I've done my replication of my DNA to make some copies, I now have two copies of this orange chromosome, this large orange chromosome. Those two copies are referred to as sister chromatids. So the sister chromatids are the identical duplicates. And we can kind of go in here and try to label these. So if I were to do that here, I've got my sister chromatids as the duplicate copies. And if I were to think about, let's say, the orange and the green, these would be homologous, meaning the same or from the same origin, and we refer to them as chromosomes. So that's what I've got inside of my nucleus. This is an organism we are doing that has two pairs of chromosomes. We, of course, have 23 pairs of chromosomes, although people sometimes refer to that as having 46 chromosomes. I prefer to think of this as having 23 pairs because we are a diploid organism. That means that when I produce sperm, those have 23 chromosomes they are not occurring in pairs because they're going to be products of meiosis that we'll talk about later. So let's take a look here at the next stage of this. Prophase is really about breaking up that nuclear envelope, and we are going to maybe form some structures that are going to help us later on. I'm going to kind of draw them as these things that look sort of like T's, and they're eventually going to migrate to the poles of the cell, and these are going to be our centrioles. And these centrioles are going to help with this kind of division process. But they're going to work their way towards the poles of the cell as we get into metaphase. So metaphase is our next step. And metaphase is a really, really simple one. It's about lining up along the equator of the cell. I could draw that equator in here as a dashed line. We wouldn't actually see it. An equator, of course, is just halfway between the poles. And our chromosomes are going to line up along that like this. So now I can have these in different locations, but in each case there's going to be a sister chromatid on either side. And the idea here is that we can form what we call spindle fibers, and these are going to help with the movement of these sister chromatids as they separate. Now, of course, we know we're going to get separation of the sister chromatids. Why do we know that? They're identical copies that were made in the S phase of interphase, with the idea here that if we double our DNA and then we pull it in half, we can have two cells that have exactly the same DNA. And if we scroll back up for a second, we can remind ourselves that mitosis, used for growth and replacement, is going to produce two identical diploid cells. It means the cells are going to be the same as each other. They're also the same as the cell that we started with. And in order to make all of this work, mitosis has to double the DNA and then split it in half perfectly so that we have two complete diploid cells. Our next stage down here is anaphase. And so if we think about how we describe metaphase, we're going to have our sister chromatids line up on the equator. And if we think about anaphase, we are going to pull the sister chromatids towards the poles. Now, we often say that this happens with the shortening of the spindle fibers. Molecularly, it's actually a lot more complicated than that, and we don't really care about those particular complications unless you happen to be a molecular biologist who works specifically with cell division. There's some interesting stuff going on there. We just want to know that in order to be able to do this, to be able to make a new cell, we need to pull on these sister chromatids. And we're going to do so from that middle point where we attach to them, that thing we call a centromere. So if we think about what's going to happen, if we pull on the centromere, it's going to get pulled first, and it's going to kind of leave its tail ends sort of towards that middle as we get them pulled off towards the poles. So what we're going to see here is the middle of the chromatid getting pulled towards the pole, and we're leaving those tails dragging behind. We do that for every single pair. Whoops, that was the wrong color to use right there. Let's see. Here we go. And you can see that this should be a pretty easy phase to identify. So here we go. We've got our completed anaphase. It's like metaphase, but we're not on the middle anymore. We're getting pulled towards the side. Now, our final stage of this is referred to as telophase. This is going to, of course, be the end of this process, and you can see that it looks like we're almost getting two cells. They just haven't completely split apart yet. So if we take a look at what we would need to do, we obviously need to finish pulling those sister chromatids towards that pole. And then, of course, to be a eukaryotic cell, we're going to have to put a nucleus back in there. And we're going to do that on both sides. So we'll draw in this reforming nucleus, and this kind of reformation of the nuclear envelope is going to be one of the key features of this. We will then take a look at what we have in here. We've got a large orange chromosome in each one, a small orange chromosome. And of course, because we have homologous pairs, we're going to have our large green chromosomes and our small green chromosomes in there with those orange ones as well. So hopefully that eventually is going to show up in here, and we're going to get a nice sync between these. There we go. All right, so we can see that we end up with what we started with. We don't have any sister chromatids in these new cells that are forming, but we do have homologous pairs. So we can say that we're going to have here two cells that have two pairs of chromosomes, and they are obviously identical to each other. Both have a large orange and a large green chromosome, a small orange and a small green chromosome. And all we need to do is to finish this process. So we finish by splitting apart in what we call, obviously, the finishing of this, or the splitting of the cell, cytokinesis, which really does just mean splitting of the cell. So that's going to be kind of the thing that follows up on telophase. And that right there is our PMAT, our prophase, metaphase, anaphase, and telophase of mitosis. Just as a reminder, of course, mitosis forms two identical diploid cells for the purpose of growth and replacement. Okay, now it's time to take a look at meiosis. As a brief reminder, meiosis is the process we use prior to sexual reproduction to produce our gametes. As a result of meiosis, we're going to produce 4 non-identical haploid cells. And again, as a reminder, we've got our math equation up there, n plus n equals 2n, meaning essentially that an egg plus a sperm equals a new diploid individual. So we're going to go ahead and take a look at this process of meiosis, and you can see here we're going to have meiosis 1 and meiosis 2. Meiosis 1 is going to start with a single cell and end, like mitosis did, with 2 cells. Each of those cells then will become the top cells in meiosis 2. That is, that each of the cells produced by the first division, meiosis 1, will enter the second division, meiosis 2, and divide, and we're going to end up with 4 cells. Now, that's not an entirely true statement. So that's one place where we need to really kind of understand that sometimes when we present something, we're going to present a standard case of this. This might be how meiosis works out in a plant. This definitely is how meiosis works out in the testes of a male. We do produce 4 sperm as a result of meiosis. But in females, things are a little bit different. We actually don't have both cells that result from meiosis 1 divide. One will end up dividing and potentially becoming the egg. The other one becomes what we refer to as a polar body. So while there is this whole meiosis process, or at least most of it in females as they're producing their eggs, from each initial cell they produce only a single egg. So the process is a little bit different for egg formation than it is for sperm formation. So we can kind of think about what we're doing is taking a look at sperm formation. And of course, we know roughly how this is going to work, right? Because we've got our acronym of PMAT, and it still applies here. So we can think about how we're going to have prophase, metaphase, anaphase, and telophase. But we actually refer to those as prophase 1, metaphase 1, anaphase 1, and telophase 1 if we're talking about meiosis 1, and prophase 2, metaphase 2, anaphase 2, and telophase 2 if we're working on meiosis 2. So we're going to kind of go relatively briefly through meiosis just to try to get an idea of how this really differs from mitosis. And let's begin first with something it has in common with it. We have a nucleus surrounding our genetic material, and we need to break that down. Now it ends up that I'm going to go ahead and jump all the way down to telophase because we know that in telophase we are going to rebuild that nuclear envelope. So we break down the nuclear envelope for prophase. We're of course going to line up at the equator in metaphase. That's going to be part of what we're going to be doing. When we get to anaphase, we're actually going to move away from that equator. So I can go ahead if I want to and prepare for that as well. So we can see that there's basically the same kind of stuff going on. Our preparations for this process are roughly the same. We know that we're going to need to have some centrioles, which form some spindle fibers, because we're going to line up along the equator during metaphase 1, and we're going to pull something apart during anaphase 1. When we get those things apart towards those poles, they're going to have a nuclear envelope form around them in telophase 1. We know that when we get to meiosis 2, we would have to break that nucleus down. And at the very end of this process, of course, we would have to form nuclei around the cells that result from telophase 2. So we've got all the same basic things going on that we had in mitosis. You might be asking yourself, what besides having two divisions is going to be different in this meiosis process? And it ends up that there are some really big differences. And one of the most important things that happens that's different occurs within prophase 1. So prophase 1 houses one of the most important things that occurs as far as preparing us for sexual reproduction. But let's go ahead and begin and imagine maybe I've got a cell that's like the cell we had before that has this large orange chromosome. And let's say that it has a small orange chromosome as well. I'm not going to draw the other components in yet, but you know that in order to have these pairs, we're going to also have a large green chromosome, and we're going to have a small green chromosome. But let's go ahead before we do that and recognize that during the S phase of interphase, we were going to create our sister chromatids as we did that duplication. That still happens here. Okay, and what I'm going to do next is that difference. And I'm going to illustrate it just between two of the sister chromatids where we're seeing a change or a difference between the sister chromatids that form that orange X or that green X. We have a crossing over, and this is actually a term we use, crossing over or recombination, between the green and the orange. That is this process of crossing over, or we have another word for this, synapsis, when they do kind of hug together. This happens between homologous chromosomes. And we can show that happening with our smaller ones as well. So we can see that there's some point at which these things cross over each other. they're actually going to do some exchange of segments in there. It's not just a simple crossing over where they kind of hug or anything like that. They actually exchange those little segments at that point, which means that we're going to keep drawing little pieces of green on one of our big orange strands and a little piece of orange on one of our big green strands the entire time that we're working our way through this. So let's go ahead and slip off to the side and talk about that a little bit. Crossing over, or what we sometimes call recombination, is going to take place during prophase, and we use Roman numerals usually, but prophase 1. Okay, it's crossing over is an exchange of segments between homologous chromosomes. And I want you to think briefly about what that really is going to mean. If we're going to have an exchange between these homologous chromosomes, we said earlier that I got one of those homologous chromosomes, let's say the orange, from my mom, and I got the other homologous chromosome, let's say the green, from my father. When they cross over and exchange segments, I'm taking the genetic material that I got from my parents, and I'm doing a little bit of blending of that. So when we take a look at how these things line up, we're going to see something unique as well, but we're also going to see that evidence of crossing over. So I'm going to start here with this large orange thing, and I put a little piece of green on it where that crossing over took place. Now you might already see the other weird thing that's happening. I'm not lined up with a sister chromatid on either side of the equator like we were when we were taking a look at mitosis. Something different is happening here. So I'm lining up with one member of the homologous pair on each side of the equator, telling me that it's actually the homologous pairs we're going to pull apart. So here I can place my large green, and then the green that has the chunk of orange on it. Now, obviously, the same thing is going to happen for the small ones, but we need to recognize something that's going on here. If we take a look at this, I could have my small orange one, and again, there would be a piece of green on it, right? On one side here, the same side as the large orange one. And I could have my other one, the one that's green but with a little piece of orange on it, on the same side as the large green one. That's one way that this could happen. But it could happen a different way. So I'm actually going to get rid of those, and I'm going to do it the other way it could happen, with this little green one that has a little tiny bit of orange on it over here with the big orange one. And I'm going to have that little orange one with a little bit of green on it over here with the big, mostly green one. So the thing that's happening here, and which happens during metaphase, is what we call independent assortment. Or actually sometimes also call this independent alignment. So independent assortment, this is the random lining up of chromosomes on the equator during metaphase 1 and 2. Now, what we need to know is that during metaphase 1, it is going to be homologous chromosomes that line up. But of course, since we're going to end up pulling those apart, we're going to have sister chromatids lining up along the equator, doing the independent assortment when we get to meiosis 2. So let me go ahead and finish up with this. We need to have some spindle fibers come in here and hook to these. That tells me that those things are going to be pulled towards either pole. It does get a little trickier to draw this. So I'm going to start off with kind of the plain thing. I'm going to add some legs in there. One of them has a little chunk of green on it. Okay, so that's what we kind of have to draw getting pulled, is going to be a 4-legged little thing here with still some evidence of crossing over. Of course, on the other side, it is a mostly green one with a little bit of orange getting pulled towards the other pole. And obviously, it gets tricky with the small ones as well. I have to remember which one I had on that side. With the green one, it's going to be the mostly orange one. But a reminder of having a little bit of green in there. And on the other side is the small one that is mostly green, but has a little bit of orange. And there I have my completion of my anaphase. Anaphase is kind of really simple. We're going to pull apart whatever lined up on the equator during metaphase. So that means that during anaphase 1, we're going to pull apart homologous chromosomes. We have this entire, homologue, the orange one, the one we got from mom, or the green one that we got from dad, and we're pulling those apart from each other. The idea here is that we're cutting our genetic material in half in preparation to make these eggs or sperm. And of course, what's going to get pulled apart when we end up getting to anaphase 2 will be sister chromatids, because we've already said that's what's going to line up along the equator. So we'll just wait for the internet to catch up with us, and hopefully we'll get that all set up there in just a moment. So that prepares us then to think about telophase. Now, we know telophase in kind of a really simple way. Telophase is just putting whatever was moving towards the pole in anaphase inside of some kind of rebuilding nuclear envelope. So that's all I need to accomplish as I start working on the telophase, is just take a look at whatever I had in anaphase and get that thing kind of moving back in the right direction. So let me see if I can work on that while we wait on this to hopefully do some updating. And I'm going to have up top this homologous pair, whoops, not with that color, that is mostly orange, but has a little bit of green on it. And then I'm going to have next to it a small version that is mostly green, but will have a little bit of orange. And then I'm going to have down in the bottom one, that large one that is mostly green, but has a little bit of orange on it, and a small one which is mostly orange, but has a little bit of green on it. Now I'm going to wait just for another little second to see if maybe I can get this thing to go ahead and update itself. Sometimes I have a little bit of trouble with that. And let's take a look and see if it's willing to give it a go. All right, and as we can see, we have down here in our nuclear envelope a single large chromosome. It does exist as two sister chromatids, and we have a single small chromosome existing as two sister chromatids. And down here in our newly forming cell down below, we have another single large chromosome existing as two sister chromatids, and a single small one existing as two sister chromatids still stuck together. Those are the things that are going to move back up to the top for prophase 2. So I'm only going to do this for one of the two cells. It ends up that there's no need to do it for both of them. It all kind of looks the same. I'm going to take the one that's in the top one. So we're going to grab that top one, and we're going to be able to move it up to that cell that is sitting there on the left-hand side. I can actually, maybe if I'm lucky, cheat and do this by selecting this thing and grabbing it, and I'm going to pull it up towards the top, and let's see if that pops up in there. There we go. I know that's kind of cheating, but it allows me to say that that's my starting point. For prophase 2, I don't do anything fancy. There's no crossing over. Let's think about why there's no crossing over. In prophase 1, I could do crossing over because we're talking about homologous chromosomes. This organism has a big chromosome and a small chromosome, and because it's a diploid organism, they exist in pairs. It received one large chromosome from its mom, one large chromosome from its dad, one small chromosome from its mom, one small chromosome from its dad, and these homologous pairs carry the same genetic information. So let's say on the large chromosome, there's a gene that controls for eye color. It ends up that eye color is not that simple. It's not coded for by just a single gene. There are very few traits that are, but we're going to treat it as if it is. Let's say that the allele, the form of that gene that you received from your mother, was for a blue eye, whereas that allele you received from your father was for a brown eye. They're both the same gene. They're coding for the same trait, but they are different alleles. And let me write that word over here by crossing over. Allele, those are an alternate form of a gene. In this case, the blue-eyed allele and the brown-eyed allele. So what we could have are different forms of genes. That's why crossing over matters, but they are the same genes as long as we have homologous chromosomes that are crossing over. And that helps explain why we don't see crossing over in meiosis 2. We know we can't see it because we don't have homologous chromosomes anymore. We have the big chromosome, but just one copy. And we have the little chromosome, but just one copy. It actually kind of makes it pretty easy to think about what's going to happen in metaphase then. We're going to have our big chromosome line up with sister chromatids on either side at the equator. Now, what I haven't drawn in here yet is the little green chunk. So what we've got going on is I know I need a green chunk, but is it on the top or is it on the bottom? Well, that's the independent assortment again, right? I'm going to go ahead and add that to the bottom, okay? But it could have been on the top. Independent assortment means we're going to have a randomization, and in this case, it's going to be for the sister chromatids. We can get the green one on the bottom, we can get the green one on the top. So as we think about what happens next with our small chromosome, which is mostly green, we have a solid green sister chromatid, and then we have the green chromatid that shows the crossing over event and has that chunk of orange on it. Well, those could occur either way. I could have the crossing over bit on the top or on the bottom. What I'm going to do is go ahead and do the solid green on the bottom, and I'm going to have that little bit with the orange chunk on it That's going to be on the top. And of course, I need to draw in some spindle fibers because I'm going to pull these towards the poles. And we can go ahead and have our Metaphase 1 all finished up there. What we're going to pull apart in Anaphase will be these sister chromatids. So this is going to look a lot like the mitosis, but we don't have full pairs of chromosomes. It's clear our organism is no longer diploid. It is haploid. And we have evidence of crossing over, which happened in Prophase 1. So let's go ahead and finish this up by taking a look at our Anaphase. So for our Anaphase, we're going to have a large orange chromosome and a large orange chromosome with a chunk of green on it. We'll have our solid green little one down on the bottom and our little green one with a chunk of orange on the top. We'll get some spindle fibers in there, pulling those things towards the poles. And once we're done with that, we can see what's going to be placed within that reforming nuclear envelope in telophase 2. So I'm going to have in this upper cell a large orange chromosome with no green on it. Down in the lower one, there will be that large orange chromosome that has that little piece of green that crossed over. In that bottom one, we have that small green chromosome. And in that top one, we have that small green chromosome that has that little piece of orange in it. Now, if we want to think about what's going to happen on the other side, for that other cell, we can imagine what would happen there. I'm really quickly going to go ahead and grab the results from that bottom cell and drag them up to the top, just as we did with the previous one. We're not going to do the work all the way through, but we can understand that we will line up sister chromatids on the equator during Metaphase 2 for this group. We're going to pull those sister chromatids apart during Anaphase 2, and that means that we're going to have a large green chromosome in one of the two cells. We're going to have a large green chromosome with a chunk of orange on it in one of them. Now, these could be different. The one with the orange on it could be in the top one. The solid green could be in the bottom one. That's, again, independent assortment occurring. I'm just going to do this in a particular way. We're going to do a solid orange, small one up top, and the orange one with a little bit of green on it will be down in the bottom. But what we want to do here is just remind ourselves what our expectation was. We scroll back up and we take a look at our expectation for meiosis. Meiosis, again, being used for sexual reproduction. We're forming gametes, eggs or sperm. We're going to come up with 4 cells. If I take a look down there, I've got my 4 cells. Those cells are going to be non-identical. All 4 of them will be different, and they're going to be haploid. Let's take a look. Well, this is the only one with a solid orange large chromosome. All the other ones have something different for their large chromosome, therefore they are all different. All of these have a single large chromosome and a single small chromosome rather than homologous pairs, so these are all haploid cells. We've just formed from a single cell 4 non-identical haploid, in this case probably sperm, that can be used for sexual reproduction.